Hello student, I am Professor Devashish Bose, Head, Department of Criminology and Forensic Science, Dr. Hari Singh Gaur Vishwavidyalaya, Sagar, Madhya Pradesh. Today, I am going to present a lecture of BSc 5th semester on the unit Introduction to Drugs, which has been jointly prepared by myself and Ms. Neha Sharma, PhD scholar, a UGC GRF at Department of Criminology and Forensic Science, Dr. Hari Singh Gaur Vishwavidyalaya, Sagar. So, let's start our discussion while taking a look at what we are going to learn today. In today's lecture, we will see Module 1, that is Introduction, Module 2, Principle of Drug Action, Module 3, Mechanism of Drug Action, Module 4, Therapeutic Action of Drug, Module 5, Conclusion. Dear students, before starting the lecture, first of all, we will try to understand what are drugs. Drugs could be the natural or synthetic compounds which causes physiological or psychological effect on the body after its intake. Pharmacological is the science that deals with the drug. This term is derived from a Greek words pharmakon, an active principle, and logos, a discourse or treat. While the term drug derived from the French word drogue, a dry herb. According to WHO, a drug is any substance or product that is used or intended to be used to modify or explore physiological systems or pathological state for the benefit of the recipient. Pharmacopoeia is an official code containing a selected list of the established drug and medicinal preparation with description of their physical property and test for their identity, purity, and potency. Pharmacopoeia also defines the standards which these drugs preparation must meet and may mention their average dose for an adult. Sources of drugs. Dear students, following could be considered as potential source of drugs or origin of drugs. Minerals, animal, plant, semi-synthetic, synthetic, microorganism, genetic engineered. Route of administration. Drugs may be applied locally or may be administered orally or by injection. Local application. A dusting powder, paste, lotion, drops, ointment, plaster is used for its action on the site of its application. Oral or eternal route. This is one of the methods used commonly for drug administration because it is safe, convenient and economical. As every coin has two sides, therefore, it also has some disadvantages. The onset of drug action is delayed. Irritant and unpalatable drugs cannot be administered. This route is useless in case of diarrhea and vomiting. Drugs which gets digested by the digestive juice cannot be administered by this route. Less absorbable drugs cannot be administered. Parenteral route. Routes of administration other than alimentary tract are called parenteral route. The advantages of this route are that drugs can be given in an unconscious or an uncooperative patients. Also, it avoids the modification of drugs by the digestive juices. It is less safe and more expensive. Also inconvenient to use. It ruptures our body tissue like artery or vein. The parenteral routes are inhalation. Drugs given by these routes are quickly absorbed and produce local and systemic effects. Injection. Drugs can be injected by following methods so that it can directly circulate in the blood. Intradermal, subcutaneous, intramuscular, intravenous, intraarterial, intrathecal, intraperitoneal, intramedullary, intraarticular. Apart from these routes, some transcutaneous, transmucosal, transnasal, transrectal and endotracheal routes are also there but rarely used for the patients. Try to understand this. Many times the term action and effect of a drug are being used as synonym, but initial consequences of drug cell interaction in action of drug while the remaining event which follow the action are called drug effects. The basic type of drug action can be classified as stimulation, It is a selective enhancement of level of activity of specialized cell. Example, adrenaline stimulates heart, but excessive stimulation followed by depression. Example, high dose of picrotoxin, a central nervous system stimulant, produces convulsion followed by coma and respiratory depression. Depression. It is selective diminution of activity of specialized cell. Example, barbiturates depressed central nervous system. Certain type of drug stimulates one type of cell but depresses other. Example, acetylcholine stimulates smooth muscles but depresses SA node of heart. Irritation. It causes non-selective and often noxious effect and is applied to less specialized epithelium or connective cells. Mild irritation may stimulate associated functions. Example, bitter increases salivary and gastric secretion. Counter irritants increases blood flow to the site, but strong irritation results in inflammation, corrosion, necrosis and morphological damages. This leads to diminution or loss of function. Replacement. This refers to the use of natural metabolites, hormones or their congeners in deficiency states. Example, levodopa in Parkinson's, insulin in diabetes mellitus, iron in anemia. Drugs may be used as replacement when the production of endogenous substance is reduced. Cytotoxic action. Selective cytotoxic action for invading parasites or cancerous cells. Attenuating them without significantly affecting the host cell is utilized to cure or palliation of infections and neoplasm. Example, penicillin, chloroquine, etc. Action of some drugs can be explained on the basis of their simple physical or chemical properties. While majority of drugs act in complex manner, the fundamental mechanism of drug action can be distinguished into 4 categories. Physical action. Physical properties like mass, adsorptive property, osmotic activity, radioactivity, radioopacity, etc. of drugs are responsible for their action. Chemical action. The drug reacts extracellular according to simple chemical action. Example, antacid neutralizes gastric HCl. Acidifying and alkalizing agents reacts with buffer in plasma and alters pH of urine. Oxidizing agents are germicidal and inactive ingested alkaloids. Chelating agents detoxify toxic metals. Through enzymes, almost all the biological reactions are carried out under catalytic influence of enzymes. Hence, enzymes are a very important target of drug action. Drugs can either increase the reaction known as stimulation or decrease the rate of enzymatic mediated reaction known as inhibition. Stimulation. In physiological system, all enzymatic activities are optimally set. Therefore, activation by foreign substance drug is unusual. Enzyme stimulation is relevant to many endogenous mediators and modulators. Example, adrenaline stimulates as adrenaline cyclase. Pyridoxine acts on a cofactor and increases decarboxylase activity. Stimulation of an enzyme increases its affinity for the substrate so that the rate constant of the reaction decreases. Inhibition. Inhibition of enzyme is a common mode of drug action. Inhibitions can be of 2 types, non-specific inhibition. Many chemicals and drugs are capable of denaturizing protein. They would alter the tertiary structure of any enzyme with which they come in contact and thus inhibit it. Therefore, they change the structure of any enzyme and inhibit the action of enzyme. Heavy metal salts, strong acids and alkalis, alcohol, formaldehyde, phenol inhibit enzymes non-specifically. Specific inhibition. Many drugs inhibit particular enzyme without affecting the action of other enzymes. Such inhibition may be either competitive or non-competitive. Competitive, that is equilibrium type. In this mechanism, drug competes with normal substrate or coenzyme, and as a result of this, new equilibrium is achieved by drug. Such inhibitors increase the rate constant, but V maximum, that is maximum rate of metabolism at high concentration of unbound drug, remains unchanged. Therefore, if substrate concentration increased sufficiently, it can displace the drug from complex equilibrium and same maximum reaction velocity can be attained. Example, cyclostigamine and neostigamine compete with acetylcholine for cholinesterase. Carbidopa and methadopa compete with levodopa for dopa decarboxylase. Sulfonamides compete with PABA for bacterial folate synthesis. Allopurinol competes with hypoxanthine for xanthine oxidase. A drug may also compete with a coenzyme. Example, warfarin competes with vitamin K, which acts as a coenzyme for enzyme synthesizing clotting factors in the liver. Competitive, that is non-equilibrium. In non-equilibrium inhibition, drug reacts with the same catalytic sites of enzyme but form strong bonds like covalent bond or have the high affinity that substrate is not able to displace drug. Example, organophosphates react covalently with the esteric site of the enzyme cholinesterase. Methotrexate has 50,000 times higher affinity for dihydrofolate reductase than the normal substrate DHFA, dihydrofolic acid. In these substitution, it is unbound drug concentration at half Vmax is increased and Vmax is reduced. Non-competitive. In this inhibition, drug reacts with an adjacent site, not the catalytic site, but it will alter the catalytic site of enzyme in such a way that it will lose its enzymatic property. Therefore, Km is unchanged, but Vmax is reduced. Example, disulfiram alters the enzymatic property of aldehyde dehydrogenase. Theophylline alters phosphodiesterase. Through receptors. Receptors is defined as a specific binding sites with functional correlates. Receptors are situated on the surface or inside the effector cells and specific agonist combined with them to initiate the characteristic response. Some drugs act by combining with ions or small molecules, but majority of drugs interact with macromolecular components of tissue called receptors. Recent studies showed that receptors are proteins or glycoproteins, and a number of receptors have now been identified and some have been cloned by genetic engineering technique. The receptors act as specific recognition molecule for ligands and as signaling molecules for activating various intracellular chemical process through second messengers. Transducer's mechanism of translation of receptor activation into functional response can be grouped into four major categories. Receptors falling in this category considered to belong to one family of receptors. G-protein coupled receptor. The largest family of cell surface receptors are the G-protein coupled receptors. There are hundreds of different GPCR proteins and nearly a third of all drug target this type of receptors. A diverse set of ligands bind to this type of receptor, including peptide hormones, neurotransmitters and other molecules. These receptors all have a similar structure with 7 transmembrane domains. On the basis of their 7 transmember domains structure, mainly GPCRs have been identified in the human genome. Proteins that were identified by sequence homology but whose ligands are not known are termed orphan receptors. GPCRs associated with heterotrimeric G protein, green, that is G protein composed of 3 different subunits alpha, beta and gamma. The subunits are tethered at the membrane surface by covalent attached lipid molecule. When a ligand binds the receptor, the activated G protein then dissociates into an alpha, G alpha and a beta gamma complex. G alpha bond to GTP is active and can diffuse along the membrane surface to activate and sometimes inhibit target protein, often enzymes that generate second messengers. Likewise, the beta-gamma complex is also able to diffuse along the inner membrane surface and affect protein activity. Inactivation occurs because G-alpha has intrinsic G-tipase activity. After GTP hydrolysis, G-alpha bond to GDP will reassociate with the beta-gamma complex to form an inactive G protein that can again associated with a receptor. The GTPase activity of the G-alpha can be made faster by other protein, sometimes the target protein itself, sometimes a separate regulatory protein. Cholera toxin caused a chemical modification that prevent GTP hydrolysis and leads to unregulated signaling. There are three major effector pathways through which G protein couple receptor function. Adenylyl cyclase, CAMP pathway, one type of G-alpha activity, the enzyme adenylyl cyclase, which catalyzes the formation of second messenger cyclic AMP. Because an activated adenylyl cyclase can generate many molecules of CAMP, this is a mean to amplify the signal. CAMP can have several effects, but a major effect is to bind to and activate protein kinase A, pKa, then phosphorylates target protein in the cell. CAMP is rapidly broken down by phosphodiesterase, limiting the length of the signal. A specific example of a receptor that couples to the type of G protein is the beta-1 adrenergic receptor found in the heart. Beta-1 receptors are located in the pacemaker cell of the heart, the sinoarterial node or the SA node, and also in other cardiac muscle. The ligand for this receptor is norepinephrine, the neurotransmitter that is released by sympathetic postganglionic neurons. Stimulation of beta-1 receptor leads to pKa activation and pKa phosphorylates membrane calcium channel, making them more likely to open. The increase in the intracellular calcium results in an increase in the heart rate and in the strength of cardiac muscle contraction. These receptors are the target of drugs, beta blockers, that are used to treat heart failure and hypertension. Phosphorylase C, IP3 DAG pathway. A different type of G-alpha activates the enzyme phosphorylase. This type of G-alpha couples to various GPCRs found on smooth muscles such as the oxytocin receptor. Phospholipase C is an enzyme that cuts PIP2, a membrane phospholipid, to generate two second messengers, IP3 and diacrylglycerol. IP3 is water soluble, diffusing through the cytosol to bind to and open a ligand gate calcium plus channel in the endoplasmic reticulum. Thus, stimulation of a receptor linked to this G-alpha is a way to increase calcium inside the cytosol. Calcium in the cytosol exerts its effect binding to calcium binding proteins such as calmodulin. In the uterus, the increase in intracellular calcium that results from oxytocin signaling cause the smooth muscles to contract. DAG is lipid soluble and stays in the membrane. It activates protein kinase C, which like pKa phosphorylates particular target protein. Channel regulation. The activated G protein can also open or close ionic channels specific for calcium, potassium or sodium ions and bring about hyperpolarization, depolarization, changes in intracellular calcium ions. Example, GS opens calcium channel in myocardium and skeletal muscles, while GI and GO open potassium channel in heart and smooth muscles as well as closes neuronal calcium channels. Physiological responses like changes in entropy, coronotrophy, transmitter release, neuronal activity and smooth muscle relaxation follows. Receptors with intrinsic ion channel These cell surface receptors enclose ion selective channels within their molecules. Agonist binding opens the channel and causes depolarization, hyperpolarization, changes in cytosolic ion composition depending on the ion that flows through. The nicotinic chlorinogen GABA-A, glycine, excretory AA and 5-HT3 receptors fall in this category. Thus, in these receptors, agonist directly operates into channel without the intervention of any coupling protein or second messenger. The onset and offset of response through this class of receptor is the fastest. Enzymatic receptors. This class of receptors themselves are enzymatic proteins. The agonist binding sites and the catalytic lie respectively on the outer and inner face of the plasma membrane. These two domains are interconnected through a single transmembrane stretch of peptide chain. Some peptide hormones and some cytokines utilizes this class of receptors. The enzyme in most cases is a tyrosine protein kinase, and the intracellular events are triggered by phosphorylation of relevant proteins. In addition, the receptors itself get autophosphorylated on tyrosine reduced, which promotes association of several receptors molecule, organizing the complex signaling mechanism. A common feature of this class of receptor is their activation and association also promotes receptors internalization and downregulation. In some cases, the enzyme is serine and threonine protein kinase. Receptors regulating gene expression. In contrast to the above three class of receptors, these are intracellular soluble proteins which respond to lipid soluble chemical messenger that penetrates the cell. The receptor protein is inherently capable of binding to specific genes but is kept inhibited till the hormone binds near its carboxy terminus and expose the DNA binding regulatory segment located in the middle of the molecule. Attachment of the receptor protein to the gene facilitates their expression so that specific mRNA is synthesized on the template of the gene. The mRNA moves to the ribosome and directs synthesis of specific protein which regulates the activity of the target cell. All steroidal hormones, thyroxine, vitamin D, vitamin A functions in this manner. This transduction mechanism is the slowest in its time course of action. The intensity of response increases with increase in dose, or more precisely concentration at the receptor, and the dose response curve is a rectangular hyperbola. This is because drug receptors interaction obeys laws of mass action according to E is equal to E maxima into D by Kd, where E is the observed effect at a dose, D of the drug, E maxima maximal response, Kd dissociation constant of the drug receptor complex. If the dose is plotted on a logarithmic scale, the curve becomes sigmoid and a linear relationship between the log of dose and the response is seen in the intermediate zone, that is, response is proportional to an exponential function of the dose. The median lethal dose or LD50. This is the dose, that is milligram per kg, which would be expected to kill 1/2 of an unlimited population of the same species and strain. The median effective dose or ED50. This is the dose milligram per kg which produces a desired response in 50% of the test population. Therapeutic index. It is an approximate assessment of the safety of the drug. It is expressed as the ratio of the median lethal dose to the median effective dose. Ti is equal to LD50 by ED50. The margin of safety is the difference between the therapeutic and the lethal dose of the drug. The larger is the therapeutic index, the safer is the drug. For safe therapeutic application of a drug, its therapeutic index must be more than 1. You know the term drug is as old as disease. Previously, different drugs were given in the form of herbal extracts while nowadays it took different form as we studied in mode of administration of drug. Drug may be exposed to our body in any form, that is solid, liquid or gaseous. It changes its structure and form and circulates to the whole body through blood. We know that if drug is given to human, it will affect positively, but sometimes negative effect is also seen in the form of different side effects. But without the drug, survival is not possible. With all these information here, we come to the end of today's interesting lecture. Do keep in mind what we have discussed today. It's time for you all to do some self-study. This is Professor Devashish Bose signing off. If you want to learn more and enhance your knowledge, you may log on to our website for MCQs, quizzes, LOR at www.cec.nic.in. Till then, goodbye.